hello guys. By the way, I'm Manalundong, and we are the group 7, and we are going to discuss to you the chapter 5, which is all about the bioenergetics. So first, I'm going to discuss to you all about the cell.
So what is a cell? In biology, it is the smallest unit that can live on its own and that makes up all living organisms and the tissues of the body. It is also the smallest structural and functional unit of an organism. It is the building block of a living organism. So this is the picture of a cell. Next is the discovery of a cell. So the invention of microscope helps scientists to study what living organisms composed of. So the cell was first discovered and named by Robert Hooke in 1665. He remarked that it looked strangely similar to a cellula or small rooms which monks inhabited, thus deriving the name. However, what Hooke actually saw was the dead cell walls of blood cells, or it is called cork, as it appeared under the microscope. So let's move on to the cell theory. So sorry if I'm wrong in pronouncing their names, but I'm going to try it. The cell theory was the first proposed by Matthias Skleden in 1838 and Theodor Schwann in 1839. Rudolf Virchow in 1855 later added the concept of information of cell. So this was the cell theory as is follow. First is the all living things are made up of cell. The second is it is the smallest living unit structure and function of all organisms. And the last is all cells arise from the pre-existing cell. So let's move on to the type of cell. So living things vary in terms of numbers of cells they have. So some living things are multicellular and other are unicellular. So what do you mean by multicellular? Multicellular is two or more cell, and other are unicellular. Unicellular is it is a one cell. So there are two broad categories of cells: the prokaryotic cell and the eukaryotic cells. So cells can be highly specialized with specific function and characteristics. So this is the picture. So let's move on to the comparison of a prokaryotic and eukaryotic cells. So I'm going to discuss to you some of these characteristics. So when it comes to size of a cell, prokaryotic typically 0.2 to 2.0 in diameter, and eukaryotic cell typically 10 to 100 in diameter. Some of the examples are in prokaryotic the example of a prokaryotic cell is a bacteria and archaea. In eukaryotic cell, it is animals and plants. So when it comes to a nucleus, in prokaryotic, nucleus is absent, while eukaryotic cell is present. So let's move on to the cytoplasm. When it comes to prokaryotic cell, there is no cytoskeleton or cytoplasmic streaming. In eukaryotic cell, there is a cytoskeleton or a cytoplasmic streaming. So the next is the sexual reproduction. In prokaryotic cell, there is no meiosis, transfer of DNA fragments only, or conjugation. When it comes to a eukaryotic cell, it involves meiosis. So parts and function of a eukaryotic cells. So the structure that make up of the eukaryotic cells are determined by specific function carried by the cell. So thus, there is no typically eukaryotic cell. Nevertheless, eukaryotic cells generally have the three components, which is the cell membrane, the nucleus, and a variety of other organelles. So next, we're going to discuss the cell membrane, or it is called also the plasma membrane. So the plasma membrane, also called the cell membrane, it is the membrane found in all the cells that separates the interior of the cells from the outside environment. So the plasma membrane consists of a lipid bilayer that is semi-permeable. The plasma membrane regulates the transport of materials entering and exiting the cell. So the primary function of the plasma membrane is to protect cell from its surrounding. It is composed of phospholipid bilayer with embedded proteins. The plasma membrane is selectively permeable to ions and organic molecules and regulates the movement of substances in and out of cells. So this is the cell membrane structure. So the next is the nucleus. The nucleus is a membrane-bound structure that contains a cell's heredity information and controls its growth and reproduction. So it is the command center of the eukaryotic cell and is usually the most notable cell organelle in both size and function. So it contains the genetic information of the cell in the form of DNA or chromosomes, and thus controls cell's growth and multiplication. This also regulates cell metabolism by synthesizing various of enzymes. So this is the picture of a nucleus. Next is the cytoplasm. So cytoplasm is a thick solution that fills each cell and is enclosed by the cell membrane. Now, it is mainly composed of water, salt, and protein. All of the organelles in eukaryotic cells, such as the nucleus, endoplasmic reticulum, and mitochondria, are located in the cytoplasm. The cytoplasm is the gel-like fluid inside the cell. Cytoplasm functions to support and suspend organelles and cellular molecules. So this is the picture. As you can see, the cytoplasm is a gel-like. So the nucleus, cell membrane, cellulose, cell wall, mitochondrion, permanent vacuole, and chloroplast is surrounded by a cytoplasm. Different organelles and their functions. So this is the guide chart. So I'm going to discuss to you some of these organelles. So first is the cytoplasm. It is both found in plant cell and animal cell. So the structure of this is clear, thick, and gel-like fluid. So the function is to hold all organelles in place, or to allow organelles materials to move around. So the next was this cell wall. It is all found in plant cell. So structure is a rigid layer of a non-living material surrounded by the cell. So the function is to support and protect the cell. So the next is the cell membrane. It is both found in animal and plant cell. So the structure is the plant layer just inside of the cell wall. Animal's outer boundary that separates the cell from its environment. So the function is to protect the cell and control the substances that go in and out of the cell. So the last is the vacuoles. It is both found in animals and plant cells. So in plant structure, one layer water-filled sac. So in animals, small round water-filled sacs, or some of the animal cells do not have vacuoles. So the function was the storage areas of cells or for food materials or waste.
Good day everyone, my name is Abdullah, and in this video, we're going to talk about photosynthesis. Photosynthesis is a process used by plants and other organisms to convert light energy into chemical energy that through cellular respiration can later be released to fuel the organism's activity. Photosynthesis is also a process by which plants make their own food. In more complex terms, it is the conversion of light energy into chemical energy by plants. This chemical energy is used by the plants for growth and nourishment. I guess you know what photosynthesis means, right? Photo means light and synthesis means putting together. So let's proceed how photosynthesis works. When humans cook food, some essential things are required, including fire, water, vegetables, and etc. Similarly, to make their food, plants also need some essential, and that's it: water, carbon dioxide, and sunlight. Water. The roots and the plants absorb the water, bring it to the leaves through tissues called xylem. Carbon dioxide gets in and oxygen gets out through tiny pores in the leaves called stomata. And sunlight. Individual photons from the sun are absorbed in the plants by pigment called chlorophyll. I have mentioned that the chemical energy from photosynthesis is used by the plants for growth and nourishment, right? So let's talk about the photosynthesis and nourishment. Photosynthesis nourishes almost the entire living world directly and indirectly. An organism acquires the organic compounds it uses for energy and carbon skeleton by one of two major modes, and that's it: autotrophic nutrition and heterotrophic nutrition. Autotrophic nutrition is a process in which the organism produces their food from the simple inorganic materials such as water, carbon dioxide, and mineral salt in the presence of sunlight. Autotrophic make their own food with the help of the method of photosynthesis. While heterotrophic nutrition is a mode of nutrition in which organisms depend upon other organisms for food to survive. They can't make their own food like green plants. Heterotrophic organisms have to take in all the organic substances they need to survive. In short, heterotrophic nutrition can't make their own food. They depend upon other organisms for food to survive. For example, bacteria, fungi, protozoan, and animals. Almost all plants are autotrophs. The only nutrients they require are water and minerals from the soil and carbon dioxide from the air. Specifically, plants are photoautotrophs, organisms that uses light as a source of energy to synthesize organic substances. Photosynthesis also occurs in algae like kelp, and certain other unicellular eukaryotes and some prokaryotes. Photoautotrophs, aside from plants, these organisms use light energy to drive the synthesis of organic molecules from carbon dioxide and water. They feed themselves and the entire living world on land. Plants are the predominant producers of food. In aquatic environments, photoautotrophs include unicellular and multicellular algae such as this kelp, some non-algal unicellular eukaryotes such as euglena, the prokaryotes called cyanobacteria, and other photosynthetic prokaryotes such as this purple sulfur bacteria which produce sulfur. On the other hand, heterotrophs obtain organic material by the second major mode of nutrition. Unable to make their own food, they live on compounds produced by other organisms, the autotrophs. So let's proceed in chloroplast. Chloroplast is the site of photosynthesis in plants. All green plants, including green stems and unripened fruit, have chloroplast, but the leaves are major sites of photosynthesis in most plants. There are about 500,000 chloroplasts in a chunk of leaf top surface area of 1 mm squared. Chloroplasts are found mainly in the cells of the mesophyll, the tissue in the interior of the leaf. So you can see at your screen the structure of chloroplast. Carbon dioxide enters the leaf and oxygen exists by way of microscopic pores called stomata. Water absorbed by the roots is delivered to the leaves and veins. Leaves also use veins to export sugar through roots and other non-photosynthetic parts of the plants. A chloroplast has an envelope of two membranes surrounding a dense fluid called the stroma. Suspended within the stroma is a third membrane system made up of sacs called thylakoids, which segregates the stroma from the thylakoid space inside the sacs. In some places, thylakoid sacs are stocked in columns called grana. Chlorophyll, the green pigment that gives leaves their color, resides in the thylakoid membranes of the chloroplast. It is the light energy absorbed by the chlorophyll that drives the synthesis of organic molecules in the chloroplast. Now that we have looked at the sites of photosynthesis in plants, we are ready to look more closely at the process of photosynthesis, and that is the photosynthesis in leaf. Photosynthesis in leaf. The main function of a leaf is to produce food for the plant by photosynthesis. Oxygen is passed into the atmosphere through stomatophores in the leaf surface. Photosynthesis green plants such as trees use carbon dioxide, sunlight, and water to create sugars. So you can see the photosynthesis in leaf structure. Photosynthesis also involves two stages, which involve a step-by-step series of a chemical reaction, and that is light reaction and Calvin cycle. Light reaction is the photo part of photosynthesis, which captures solar energy and transforms it into chemical energy, while Calvin cycle is the synthesis part, which uses that chemical energy to make the organic molecules of food. Let's proceed in light-dependent reaction. Light-dependent reaction is the stage that captures energy of sunlight, storing it as chemical energy into two different energy carrier molecules, ATP and ADPH. The chemical energy stored in these molecules will be used to power the synthesis of high energy storage molecules like glucose during light-independent reaction. As the term implies, light-dependent reaction can take place only in the presence of light, solar energy. The light-dependent reaction takes place in the thylakoid membranes or grana of the chloroplast. The thylakoid membranes contain highly organized assemblies of proteins, chlorophyll, and photosystem. Light-independent reaction, or the Calvin cycle, is the stage that uses the energy to make carbohydrates, or so-called the sugar-building phase. This process was named from the fact that they do not require light to take place. The Calvin cycle is named for Melvin Calvin, who along with his colleagues James Basham and Andrew Benson began to simplify its steps. The carbohydrate produced directly from the Calvin cycle is actually not glucose, but a 3-carbon sugar. The name of this sugar is the glyceraldehyde triphosphate, or the G3P. For the net synthesis of one molecule of G3P, the cycle must take place 3 times, fixing 3 molecules of CO2, 1 per turn of the cycle. Calvin cycle is divided into 3 phases: carbon fixation, reduction, and regeneration of CO2 acceptor. Calvin cycle is anabolic, building carbohydrates from smaller molecules and consuming energy. Carbon enters the Calvin cycle in the form of CO2 and leaves in the form of sugar. The cycle spends ATP as an energy source and consumes NADPH as reducing power for adding high energy electrons to make sugar.
Cellular respiration is a set of metabolic reactions and processes that take place in the cell of organisms to convert chemical energy from oxygen molecules or nutrients into adenosine triphosphate and then release waste products. So, it means it works with or without oxygen, but essentially, the process is called cellular respiration dahil ang cell is nagre-respire siya in a way na ginagamit ang molecular oxygen as its electron acceptor and release carbon dioxide as its end product. Now to proceed, there are three stages of cellular respiration process. These are glycolysis, the Krebs cycle, and electron transport system. The first stage, glycolysis, is the metabolic pathway that converts glucose into pyruvic acid. It literally means a splitting sugar. Glycose comes from the Greek word sweet and lysis means to split. Glycolysis is a series of reactions that extract energy from glucose by splitting it into two molecules of pyruvate. Just to be clear, glycolysis occurs in the cytoplasm outside of the mitochondria. So, during this stage, two ATPs are produced and NADH is released for a later on step of cellular respiration. Then, the pyruvate of glucose move into the mitochondria where they are converted into acetyl-CoA. During this conversion process, more NADH is produced and CO2 is released. Now let's proceed to the next stage. The citric acid cycle, or also known as the Krebs cycle, is a series of chemical reactions to release stored energy through the oxidation of acetyl-CoA derived from carbohydrates, fats, and proteins. Krebs cycle can be described as metabolic pathway that generates energy. This process happens in the mitochondrial matrix, where pyruvate has been imported following glycolysis. So, during this stage, the remaining carbons from the initial glucose are oxidized, releasing CO2. NADH and FADH2 are also produced, in addition to two ATP molecules. After that, the next stage is the electron transport system or electron transport chain. It's a series of protein complexes that transfer electrons from electron donors to electron acceptors via redox reactions and couples this electron transfer with the transfer of protons across a membrane. Now, where do you think oxygen is used in cellular respiration? In this stage involving electron transport chain, the electron transport chain is the final stage in cellular respiration. It occurs in the inner mitochondrial membrane and consists of several electron carriers. The purpose of the electron transport chain is to form a gradient of protons that produces ATP. It moves electrons from NADH to FADH2 to molecule oxygen by pumping protons from the mitochondrial matrix to the intermembrane space, resulting in the reduction of oxygen to water. Therefore, the role of the oxygen cellular respiration is the final electron acceptor. Flash forward and now, if we add up all the ATP produced from every stages in just one glucose molecule, we can see that these reactions produce a total of 36 ATP. And that's the end of it. But what if the final electron acceptor is not oxygen? That's when anaerobic respiration takes place. It is a respiration using electron acceptors other than molecular oxygen. An anaerobic type of respiration is carried out chiefly by anaerobic organisms, example anaerobic bacteria, that use certain molecule as electron acceptor instead of oxygen.